Hey Mustangs, in these notes we're going to take a look at DNA replication. Now before we start the notes, I want to make this very clear. This is a super simplified look at DNA replication. So if you were to take an AP class or a college class, it's going to be a lot more complex than this. Now we're going to start off simple, and I'll show you guys the more complex DNA replication later on in class. But for now, we'll just focus on the simplified, and this is all you'll be responsible for in my class. So again, it's super simplified. There's actually a lot more to DNA replication than what I'm going to show you right now. So just remember, AP class, college class, you would have a lot more to it. So it's going to be super simplified. All right, so first, before we start this, I want you to think about what replication means. If you think about the word replication, to replicate something means to make a copy of it. Okay, so in DNA replication, it's about making a copy of DNA molecules. And if you think about it, when do we want to make copies of DNA? Before a cell's going to divide. So if you have a cell and it's getting ready to divide and create two new cells, you want to make sure that both new cells have an exact copy of the DNA instructions. So that's when DNA replication is going to occur. You're going to make a copy of the DNA before a cell divides. So that way when a cell divides, one copy goes here, one copy of the DNA goes here, and you have two identical sets of instructions, one in each cell. All right, so the big thing you want to think of is we start with one copy of DNA instructions. Okay? Our original copy came when the sperm fertilized the egg and it created our very first cell. And from here to being an adult, we have trillions of cells. Okay, so how do you go from one copy to having the exact same copy in every single one of your trillions of cells as an adult? That's DNA replication. So here are the basics. So go ahead. Remember, when you're doing YouTube notes, make sure you pause and copy the information down, and then I'll continue talking about it. So go ahead and pause and then come back. All right. DNA replication is the process of making an exact copy of the DNA. That is the goal of replication, to make an exact copy of the DNA instructions. It's not always perfect though. So there are mistakes that are made. Those are called mutations. But for the most part, if DNA replication is going down right, it should be an exact copy of the DNA molecule. As I said in the beginning of the notes, replication only occurs before a cell is going to divide. So three types of division we see are mitosis, where you make two identical cells, meiosis, which is making sex cells, sperm and egg, and binary fission, which is how bacteria reproduce, where you have one bacteria grows bigger, makes a copy of DNA through DNA replication here, and splits into two identical, both having the same DNA instructions. Okay, so make an exact copy of DNA before a cell is going to divide. Again, make sure that both new cells started with one copy, make a copy, one copy goes here, one copy goes there, both have instructions now, exact same instructions. All right, so again, here we start with the sperm and the egg. So the sperm fertilizes the egg, gives you your first set of instructions, but then that cell, which we call the zygote, so this is the fertilized cell, which we refer to as a zygote, and we'll be learning that term when we get back from winter break. It's going to divide and form more cells. So here we see two cells. There's one cell there, one cell there. When this zygote divided to form two cells, it had to make a copy of the DNA instructions through DNA replication. So you make a copy of the instructions, one copy goes here, one copy goes there. Now both of these cells have the exact same DNA instructions inside of them. Well, these two cells are going to split, then you have four cells. Okay? Again, before each of these two cells split, this cell made a copy of instructions, and when it split, one copy went here, one copy went there. This other cell, before it split, made a copy, one copy goes here, one copy goes there. So same thing here. So each of these four cells are going to make a copy of the instructions before they divide to form 8 cells. Each of those 8 cells will make a copy of the instructions before they divide. One copy goes here, one copy goes there, to make 16 cells. Okay, eventually cells keep on dividing until you get what we call a blastocyst, and this is a whole bunch of cells all together. Okay? Now, somewhere around here is actually when the cells began to differentiate, but that's not really what we're focusing on. We want to focus on the DNA replication, making copies, making sure you make an exact copy of the DNA as they split. Okay? But cell differentiation does happen within these first few steps, where you start to actually get cells starting to change to become a specific type of cell in the body. Get ready, really, is what happens during these few steps. All right, so here's the simplified. So go ahead, pause the video, and write down the instructions or the information. All right, so super simplified. There's actually a lot more steps to this. So step 1, we're going to say a helicase enzyme unzips the 2 strands. Step 2, DNA polymerase enzyme adds new nucleotides to the old strands. Step 3, the polymerase enzymes are released and you wind up with 2 identical DNA molecules. Now we're going to use hand signals to help us remember these things. Okay, so we're going to use 2 hands to represent the 2 strands of DNA. So you have both strands of DNA. Step 1, helicase enzymes unzip the 2 strands by breaking the hydrogen bonds. Step 2, DNA polymerase adds new nucleotides to the old strands. Step 3, polymerase releases and you wind up with 2 identical DNA molecules. And that's the simplified steps of DNA replication that you're going to have to know. So here is a version of that happening. So here you start with 1 DNA molecule, and when you look at the end you have 2 identical DNA molecules, and then these are the steps in between. So helicase would go through and break the hydrogen bonds between the 2 strands. DNA polymerase would add matching nucleotides based on the base pairing rules to the old strands. And when it's all done, when it goes all the way up the entire DNA double helix here, you wind up with 2 identical DNA molecules. Now this type of DNA replication has been called semi-conservative because, and this is the key here, in each molecule there's 1 original strand and 1 new strand in the new molecules. So if we go back here, you can see these are the original strands from the original DNA molecule, and here's new strands being added to them. And when you look at the final 2 DNA molecules at the end here, there's 1 original strand that's in white and 1 new strand that's in green in both of these. So that's why we call it semi-conservative. All right, so let's go ahead and take a look at our paper model, looking at our super simplified DNA replication. So I'm going to go ahead and take a look at the paper model now. All right, so if we look at the paper model, step 1, helicase unzips the 2 strands. Step 2, DNA polymerase adds new nucleotides to the old strands. So we're adding new nucleotides to these old strands here. And if we keep on going, nucleotides being added by DNA polymerase, you wind up with 2 identical DNA molecules. Okay, so if you actually looked at them, the sequences of A's, G's, T's, and C's would be the same. So it's AT, CG, AT, GC, GC. So 2 identical DNA molecules were made.